Acho que isso tá bom.
Good morning, everyone. Good morning. We will make this opening in Portuguese. So those that don't understand Portuguese, please put on your headphones. Bom, bem-vindos. A gente vai ler um texto, tá? A gente vai ler um texto. A gente tem uma cola aqui.
We are going to read a text. We have some notes here. Dear colleagues, I'm very joyful to welcome you all to our national meeting for malaria research. We are very proud to see that this is the greatest and more traditional malaria congress that is happening in an endemic country in this 18th edition. And we have the number of the participants in this Congress in a continental country as ours. And we have 300 people with the same purpose, to create a dialogue space between institutions and experts from different fields, all committed with facing malaria. Our first National Malaria Research Meeting happened in 1986, 40 years ago. The founders of this event noticed that the efficient control of malaria depends on dialogue and collaboration of multiple actors, depends on basic science, clinic research, industry, third sector, and primary attention in the formulation of public policies. And after a while, that has changed the character of this meeting. That is a cross-cutting meeting that is integrating several fields of knowledge. And before we officially start with our program, we need and we want to thank from the bottom of our heart those who made it possible to have this meeting. First, our financial supporters, the control program, and we would like to thank Alex, FAPESP, CNPq, CASP, USP, Unifest, Fiocruz, IOC graduation program at Unifesp and USP, Institute All for Health, Gates Foundation, OGHS, MFV, Butantan Institute, Elsevier, Clark, Sumitomo, and Wood, GSK, Abbott, SBPZ. Thank you all for your partnership and your support. They're all there. And we want to thank some of you as well. We shouldn't forget about Professor Claudio. Claudio.
Muito obrigada por
Thank you so much for creating this meeting and for being with us since then. We wanted to thank you and also our partners in this organization, Beto and Josué. You worked hard for us to be here today. Gerdi, for your help with the bus. Gerdi organized the bus. Cássio, where's Cássio? Delio, Marcos, who couldn't come, unfortunately. They were key for finding resources. And we would like to especially thank Chris. Where is Chris? You don't know about that, Chris, but we were almost drowning when you helped us. And we wanted to thank Fabio Costa, who trusted us with this mission and was always available to give us valuable advice. And we would like to thank Marcelo and Lucia, who led the scientific community, and we'd like to thank all the board members for their work. And we still have many tasks ahead of us. We would like to thank all the evaluating commissions, right, Danny? Thank you all if you are part of this judging commission, and we are going to have Hutchesvine medal and best poster. And all the commissions are plural, respecting criteria and diversities. We have a very hard mission ahead of us. And other important pieces of information. We have great awards, and no organizing member, myself, Carol, Beto, Josué, we are not part of the judging board. So please do not exert pressure on us. And I'd like to introduce to you three people who are going to help us to maintain the program within time and space. So Farinha, Hugo, and Gui.
Muito obrigada. A gente tem
Thank you
certeza
so
que a
much. We are
presença de vocês
sure
vai ser
that
bastante
your
especial.
help and your presence is going to be key. If
If
the moderators and speakers, if you have doubts about the tables and the progress, they are going to guide you.
E temos mais três agradecimentos
And we are going
especiais.
to have three other
A Vera
thank yous.
Bittencourt, nossa
First,
diretora,
Vera
vai estar aqui.
Bittencourt from ML
Ela vai estar aqui,
Events.
provavelmente
Where
ela
is
deve estar
she?
por aí
She
trabalhando.
must be around working.
Sem a Vera, nada disso seria
Without
possível.
Vera,
Temos quase
nothing
certeza que
would
ela
be
vai precisar da
possible
gente
here.
depois desse evento.
We are going
Mas
to,
ela aguentou
she's going to
graciosamente
escape us
8
after
meses
this
todos
event,
os
but
dias
she
com a gente,
has
todos os dias.
endured tens of messages every day that we sent to her. Vera was the person that was asking you to enter so that we could start this meeting. So any problem that we might have, Vera will help us. So please look
E a
for
gente
Vera.
também quer agradecer a
And
todos
we
os
would like to
inscritos
thank all
do
those
grupo de
who
trabalho
registered,
e a todos vocês
everyone
que vieram de
who
perto e de
submitted
longe
works, and
e
you
agora
who
estão
came
aqui.
from far and
Desejamos que
close
nos
and are
próximos
now
dias
are
a gente
here.
possa ter
And we
sempre
hope that the four
novidades,
next days are going to bring quality
novidades,
science
novas
for
memórias,
you
novas
and
colaborações,
from you,
novas
with new
amizades,
memories,
experiências.
collaborations, and friendships in science and
Finalmente,
life.
nossa
And finally,
agradecimento muito
our thank
especial
you
à nossa
that is
presidente
very
Dilma.
special to our President Dilma,
E aplausos.
and our honor President Dilma. Please stand up, Dilma. Irene.
É um super privilégio ter você
It's
como
a
presidente
super
de honra
privilege
aqui
to have you
presente.
as a President of Honor, as a speaker for this
Essa é a
opening
inscrição.
conference. And from this
Ela se chama
conference on,
Conferência
it's
Vitor
now
Nussenzweig.
called
Vitor
Vitor
e Ruth,
Nussenzweig
pela
Conference.
Nussenzweig, passam a
So
ser
Vitor and
homenageados
Huti
na
are
nossa reunião.
always going to be honored in
Esperamos
our
que esse
meeting.
evento
And we hope that this
possa ser um
event
dia
that
muito
is going to
simbólico e
start in
significativo
this
para
very
nós,
meaningful day
April
for all of us,
25th, que é o Dia
April
Mundial de Luta
25th,
contra a
that is
Malária,
the
gerar
National
muitas
Day
e
to
fortalecer
fight
o nosso
against
compromisso com esse
malaria,
vírus.
will generate many ideas and reinforce our commitment with this disease. And we would like to give the floor to Professor Aluisio. He's going to introduce Professor Irene, and he will officially open this meeting. We will introduce now Aluisio.
O professor Aluisio é hoje
Professor
reitor da USP.
Aluisio is the dean of USP, the University of São Paulo. He's a doctor in infectiology and an expert in tropical diseases. I could read his long resume, but we wouldn't have time today to talk about all his achievements. But we are going to tell this story. 10 years ago, I started uh, teaching at USP, and the department has put me to teach on infectious diseases at USP. I was so scared because I was not a doctor. I didn't know anything about clinic, and I only knew about the parasite. And then Marcelo told me, you only have to talk about parasite because during the class you're going to have a clinician, a pathologist, an epidemiologist. And the first class I had given was about malaria, and the clinician was Aluisio in the class. Until today, that class was the best class I've ever watched. He's not only the USP dean, he's not only a doctor and infectiologist, but he's also working with malaria, and he's an excellent professor. Eh, We are so proud to have you as a dean. Thank you, Aloysio. Thank you.
Bom dia a
Good
todos.
morning,
É com
everyone.
grande satisfação
I'm very pleased to greet the Professor Daniel Bargieri, Ana Caroline Aguiar, the presidents of the ACEN National Malaria Research Meeting. I'd like to thank you for the honor to introduce the opening remarks of this wonderful scientific event. I'd like to greet the undergraduate and graduate students, the postdoctorate students, and the researchers that are now making this meeting more and more rich. I'd like to share my joy to be in this opening remarks, and it's a unique opportunity that was given to me and enables me to meet again so many friends. I'd like to say that I was present, right, Professor Claudio, in the first meeting 40 years ago, and now I'm back. The joy is even greater because we are now living this very special moment. We are celebrating the memory of Professor Victor Nussenzweig. We are going to show a slide, the joy of celebrating his memory, who is naming the opening conference of this meeting. One of the greatest Brazilian immunologists who was a highlight in the development of vaccines against malaria. There's no doubt that Professor Victor Nussenzweig is a personality that deserves all honors, not only due to his scientific and academic legacy, but also due to his ethic stance marked by the commitment with democracy, social justice, health, and the dignity of vulnerable populations. A son of Jewish Polish immigrants, he was raised in a home that was valuing education and science. Living in the neighborhood of Bom Retiro, we had three scientists who are very famous: Victor and his brothers, the Dr. Israel, and the physicians Hertz Moisés Nussenzweig. However, as Victor always said, he attributed to Ruth, his colleague in the medicine school at USP, his awakening for scientific life. It's impossible to talk about Victor not talking about Ruth. During the undergraduate course, Victor and Ruth started their studies with the trypanosoma crucis, guided by the expert of IMUSP, Professor Samuel Barsley Pessoa, an icon of biomedicine in Brazil, and we all know him very well. In one of his first of more than 500 scientific publications, the Nussenzweig couple has shown the capacity of the dye gentian violet to kill the parasite, showing its use for the transfusion of treatment of Chagas disease. Aligning the academic life to their life projects, Victor and Ruth decided to get married in 1952 during their medical studies, and he chose an unique place, the library of the medicine school at USP. After finishing his doctoral studies, Victor went to this internship at Pasteur Institute in Paris with his wife, and however, after coming back to Brazil, they've seen adverse conditions in the political scenario after the military coup in 1964 that prevented them of staying in this country. He was an activist since he was very young, and noticing the persecution that fired and arrested many of his colleagues in the Department of Parasitology in the College of Medicine at USP, he had no other choice. Victor was able to get a scholarship at Guggenheim Foundation and opted to leave Brazil with Ruth towards New York. In the medicine school at New York University. The institution that hosted him started working with the Venezuelan immunologist Bahous Emanasef that would be awarded with the Nobel Prize of Physiology in Medicine in 1980. Victor has built an international career there and became the chair and world reference in his field. Later on, when he was married with three kids and a long careers in NYU, that the Nussenzweig couple has given its greatest contribution to science by identifying this cytosporozoite protein in the infectious part of the plasmoid. And after showing that neutralization with antibodies would reduce the infection capacity of the spirochete, they established the basis for the first vaccine against malaria recommended by the World Health Organization decades later. So this discovery has transformed the fight against the disease and has opened the way for new generations of drugs. Over their successful career at New York University, Victor has advised tens of Brazilian students and Latin American students in his research group, contributing for the excellent training of generations of researchers. Many of them could be here today in this session. The couple, Nussenzweig, has established themselves in 2012 in Brazil again, having worked for a few years at Unifesp with in a partnership with Sergio Sheikman, characterizing essential enzymes for development of the plasmodium. In a testimony for FAPESP magazine, Sergio Sheikman said that Victor knew deeply immunochemistry by trying to understand the molecular issues in the malaria response. He contributed for Ruth and her team of investigators to explore innovative way to face this parasite, establishing what Sheckman called the scientific synergy that was perfect. Over his career, Victor has received many honors, like the National Merit Award in 1938 and Carlos Finley Award in 1985, besides being a member of the Brazilian Academy of Science and the Academy of Arts and Science in the United States. Victor Nussenzweig has shown us that science could be a way of resistance, and especially a way to save millions of lives. He could barely imagine that up until now we have science as a field of resistance. We are living that today. And it's undeniable that the Victor's discovery and the results of his partnership with Ruth are still relevant and considered a solid base for the advance of this drug against malaria. According to the trail that was opened by Victor and Ruth, Brazilian researchers led by Irene da Silva Soares are advancing today in the tests of a vaccine against this Vivex plasmodium, a parasite that is responsible for the most prevalent form of malaria in Latin America. The 18th National Malaria Research Meeting has the great privilege of counting to the honored presence of Professor Irene Soares for the opening conference. Uh, Graduated in medicine in the Federal University of Pará in 1983, master in microbiology in the Federal University of Minas Gerais in 1991, and doctor in microimmunology from the Federal University of São Paulo in 1998. Professor Irene is the chair of the clinical and toxicological department in the University of São Paulo. Her academic lead is filling us of pride and is translated in her work in different periods as the coordinator of the graduation program in pharmacy and toxicology in the pharmaceutical school, president of the graduate uh, studies chief of a clinical analysis in that same institution. And nowadays, the Professor Irene is now collaborating intensely with the management of the University of São Paulo. And on behalf of our university, I'd like to thank you, Professor Irene. Working as the representative of the congregation of pharmaceutic congregation in the University Council, and she's also working as a member of the curator board of the University Foundation of our exam to enter university, FUVESP. She's also a member of National Institute of Science and Vaccine Technologies that is approved between the Federal University of Minas Gerais, René Rachu Institute from Fiocruz, Minas Gerais, and the Technological Park of Belo Horizonte. I believe that we all share the same understanding that it's a great privilege to host Professor Irene Soares, whose dedication on malaria vaccines is representing the continuity of the legacy that is started by Victor Nussenzweig. Welcome, Professor Irene. Thank you. Good morning, everyone. First of all, I'd like to thank the organizing committee, especially Carol Aguiar, Daniel Mageri, and Roberto Barros, and our colleagues in the Scientific Commission. It's a great honor to me to occupy this position due to indications of my colleagues, and I deeply appreciate this event. I've been participating since 1998 with rare absences in this event that is happening every 2 years. And that means very much to me as well. It's very meaningful that I'm being honored in this event. Basically, I'm going to thank the words of Professor Aluisio, our magnificent theme, and he said a lot of what I was going to talk about in my introduction. So I'm going to be a little bit faster with the presentation. So I'm going to talk about this Brazilian vaccine that we are developing against Plasmodium vivax in partnership with the Federal University of Minas Gerais. I'm going to talk about this trajectory and up to which point we went. Since I am the first to speak in this event, I can talk about epidemiology. Just to draw the attention to the importance of malaria according to data of the yearly report of the World Health Organization. We had 282 million cases registered in 2024. From them, 80,000 deaths. Most of them were children less than 5 years old. And the Plasmodium vivax is the second most prevalent species after Plasmodium falciparum, and we had 15 million cases this year, and the population living in risk areas is up to 2.5 billion. So this map is showing that malaria is present in all the tropical and subtropical regions in the world, and we see the great concentration of Plasmodium falciparum in Africa, where we have most of the deaths. And in yellow, we show the distribution of the other species, vivax, malaria, Bergame, knowlesi, and knowlesi is a monkey Plasmodium that is called the fifth species of Plasmodium that is infecting man. Basically, what I'm drawing the attention here is that outside of Africa, the Plasmodium vivax is the species that is prevailing in the world. Here is, you see the situation in the Americas according to the Pan American Health Organization. We have 17 endemic countries highlighted in orange. In 2024, we registered more than 500,000 cases, and we have a high prevalence of the Plasmodium vivax, more than 60%. So we have Brazil, Venezuela, and Colombia with 80% of all cases.
Here I show you the conventional strategies to control malaria, the early detection of the cases, the use of insecticides in the nests of the mosquitoes, the use of anti-malarial pharmaceuticals, and the use of insecticide in the impregnated mosquito nets. The issue is that in the last few years, we have seen an increase of the resistance of the parasites to pharmaceuticals, of insecticide resistance among the mosquitoes, and the climate change that tend to hamper the control of malaria in the world. So in the World Report from 2025, in its cover, it already draws the attention to pharmaceuticals resistance. There are several elimination programs on behalf of the World Health Organization that launched this program in 2015, PAHO also in 2024, and the Brazilian government in 2022 launched a program to eliminate malaria. So our work is inserted in this context as another alternative to be combined with the other strategies so we can truly achieve these goals. The development of vaccines has been considered a priority. The Plasmodium life cycle is extremely complex. There are several different stages that are developed in the human being: the pre-erythrocytic forms, the asexual blood forms, and the sexual blood forms. Due to this cycle, a vaccine against malaria is not going to work in only one of those stages. That is why we have the concept of a multi-antigenic vaccine. And we show here that this antigenic repertoire is very extensive. Some antigens are species-specific, and that hampers the development of vaccines against malaria. Most of the vaccines are included here. These are pre-erythrocytic vaccines with the target of a few proteins of the parasites, especially the circumsporozoite protein, vaccines using the whole sporozoite, and other vaccines, most of them focused on this stage, which is the initial stage of the infection. So these vaccines aim at neutralizing the sporozoite in its in its entrance door. There are two vaccines recommended by the World Health Organization, RTSS, approved in 2021. It is for children above 5 months old in a scheme of 4 doses and an efficacy of from 30 to 50%. This vaccine is not available globally. It is being implemented in regions with moderate and high endemicity with the purpose of reducing children's mortality, childhood mortality. So this efficacy of 30 to 50% initially may seem low, but 1/3 of the children can be saved. In 2023, another vaccine also based on the CS protein, the CSP, actually it's an RTSS, an improved RTSS was recommended in 2023. and it also encompasses this age group with the scheme of 4 doses, and there was an efficiency in this case of 70%. Both of them are still in the phase of implementation, and possibly these vaccines will not be used here in Brazil and in the Americas because here Plasmodium falciparum is not as prevalent as the Vivax, and our area is not considered a high endemicity area. As Aluisio mentioned, up to 2021, approved by WHO, the first works were published by Dr. Ruth in 77 that showed that the radiated sporozoite can work against malaria using rodents. It was published in Nature, the study, and in the subsequent years they identified the protein, which is the circumsporozoite protein. They identified the region of the protein that was a target of protective antibodies. The immunodominant region, as we call it today, and also the mechanism. So that was a discovery that opened new paths for us to get to this stage of the Plasmodium falciparum vaccine, reminding you that this vaccine is not effective against the other species of Plasmodium. So our goal is to develop a vaccine against Plasmodium vivax. Vivax is considered a neglected parasite still. That is why it is given less attention. There were already 130 vaccines tested against Plasmodium falciparum or are under tests. In the case of Plasmodium vivax, only 11 vaccines were tested in human beings. That means 8% of the total. And as we can see here on the left, the pointer is not working. Here on the left, I included a comparison of Plasmodium vivax and the Plasmodium falciparum regarding the stage of each one of these vaccines. In the case of Plasmodium vivax, no vaccine went over phase 2. Some of them reached phase 1 and 2, but they did not get to advanced stages of testing. And Plasmodium falciparum, as I said, it has 2 vaccines recommended, and in total, most of them reached phase 3, and 2 of them reached phase 4. So this is just to show you the context that Plasmodium vivax receives less attention, but it is a more prevalent species in Brazil, in Americas, in Asia, so it's important to develop a specific vaccine for it. Our target is the circumsporozoite protein. We use a lot of platforms. The platforms that is most advanced is the vaccine of a protein subunit. We also use chimpanzee adenovirus and adeno5 for clinical trials, and more recently mRNA vaccines that I'm going to show you briefly. But my focus is going to be where we got to with this recombining protein platform. We know that the adjuvant has a critical role in protecting against malaria. Throughout the years, we tested different adjuvants, aluminum hydroxide, TLR agonists, oil and water, or water and oil, trying to find the best formulation. Differently than the target of the Mosquirix vaccine against the falciparum, our goal is adults, residents in endemic areas, or travelers. Most of these residents are workers in mines, or they work in rural settlements, and also indigenous populations. These are the most affected populations in Brazil. So I'm going to show you the design of this protein and the proof of concept that was developed in the University of São Paulo at the time in partnership with UniFest. Here I'm showing you the structure of the protein. There are two regions, and in the center there's this repetitive region, which is the immunodominant region of the protein. And based on this region, there are three allele variants. VK210 is the most prevalent form in the world, as you can see here in this review on the right, done by Ricardo Machado, where he shows in blue that it is comprehensively prevalent in the world, and in second place, VK247 and Vivex-like. We don't know the prevalence very well because it's not even typed in some countries. But when we look at Brazil, we see that we have the three variants, and they also occur in mixed infections. In Guyana as well, we have data showing the prevalence of Vivex-like. So we understood that it is important that this vaccine covers this whole diversity. So this study started in the Federal University of São Paulo, a work coordinated by Professor Mauricio Rodrigues in partnership with Ruth and Victor Nussenzweig. At the time, Victor was here with Sergio Schenkman, and he was also part of our theme group from 2011 to 2014. They wouldn't come to Brazil twice a year, despite their advanced age. It was the motivation of their lives, and they always have had a lot of focus. Now let's invest in Vivex, let's create a vaccine for Vivex. So he participated in the whole conception of this vaccine. Initially, we did it with 3 recombinants. Here on the left, we have a 210 Vivex-like, and we use this recombining proteins mixed, combining it with another recombining protein, which is the living Vivex, and we covered the 3 variants, covered the whole diversity of the CS protein. This work that was published in 2014 showed that using 3 proteins mixed or a single chimera protein had very similar results in terms of immunity. So if there are similar results, the advantage is to continue with the chimera protein because, of course, of the cost that takes for us to produce each one of these proteins with best manufacturing practices in a lab. We did not have a challenge at this time, but that was an important information. This was an insoluble protein. It had to be boiled and treated with urea. Thinking of the scaling, we aborted this idea of moving on with this E. coli recombinant, and we followed a very similar approach in the yeast express protein, which is the delivery form of the vaccine against falciparum. So we created this chimeric construction containing the 3 variants. And regarding the previous construction, we did not include N-terminal. There was no justification for it to be present. The only important epitope is in this region here, the R1, which is a combination region between the N-terminal and the repetitive region. This epitope has been described as the target of protective targets, and it's highly conserved among different species of Plasmodium. So we maintain this epitope, and we have the sequence of the 3 variants and the C-terminal region, which is a conserved region, and it has epitopes for T cells mapped in this region. Here we have the yeast expression system. It is a secreted yeast expression system. This makes it a lot easier for us. We go through several stages of purification, and we ended here with a highly pure product with high yield. There's the appropriate folding and the facility to scale this up with not so high costs. The first trial that we did, the proof of concept, was in collaboration with Rogério Amino from the Pasteur Institute. We did the immunizations, 3 doses. We used as adjuvant poly-C, a double band RNA agonist of TL3, and we showed here that the 3 variants were acknowledged. So the question is, if I immunize a hybrid protein, is it going to produce antibodies against the 3 variants? Yes. So we were able to immunize against the 3 variants and for the C-terminal region, which is a conserved region, and that induces antibodies. For these tests, we use proteins here that contain only the repetitive region sequence or NC terminal without the repetitive region. When we look at the isotopes of induced antibodies, we see a mixed response, a predominance of GG1, but also a good levels of GG2B and GG2C. So it's a balanced response, and these antibodies were also capable of recognizing the sporozoite here in the native protein. With this data, the issue is poly-C is not used for human vaccines. It was already tested, but it is far from being licensed. So we started testing other adjuvants, including here aluminum hydroxide, a comprehensively used adjuvant in vaccines. It has been acknowledged for decades, and it is licensed for use in human beings. So we did this comparison in terms of magnitude of the induced antibodies response and in terms of the longevity of the response. You can observe here that both induced high levels of antibodies, and starting from the 150, the response starts decreasing, but it is maintained in a very good level up to almost 500 days. So the issue of longevity is something very important when we develop a vaccine. Then we looked at the response of B cells from the spleen after the 2nd and the 3rd dose, and we saw that both vaccines were able to induce cells that secrete antibodies, especially plasmacytes. And when we look at after the 3rd dose at the comparison, we see that poly-C induced a much higher prevalence of antibody secreting cells than aluminum hydroxide. Here we have precursor cells. We saw that the actually here it's after the 10th day, and we analyzed in the 5th day after the 3rd dose, and then the 10th day, and we saw that the response with poly-C achieved a higher population of cells. We also looked at memory cells. We characterized these memory cells using the spleens of the mice immunized with the same adjuvants, and we saw that the memory cells and the precursor memory cells are induced more efficiently using the poly-C adjuvant. Then we moved on to the crucial trial, which was the challenge of these animals. After 3 doses, they were challenged with, since Vivax does not infect mice, we use a transgenic parasite expressing the repetitive region of Plasmodium vivax, and these animals are followed from the 4th to the 10th day. This whole protection effort was done in partnership with Dr. Rogério Amino. In the 5th day of the infection, we see a very significant difference of the control group, the group that received the protein. Here we can see 4 animals that received sterile protection. We looked at the parasite load in the liver, and it's also a very significant difference when compared to the control group. And when we look at the survival curve, these animals, 76% of these 4 animals survived until day 10.
We also used another challenge system that was established at Oxford University by Dr. Arturo Sandoval. If the same immunization system, 3 doses, and the difference is that the challenge is endovenous. So with Rogério menu, it was with the skin, and here is in intravenous. So we had the opportunity with this group to evaluate the protection with the 3 variants. So we see that we see this delay of the parasite development in the pre-patent with VK210 and Vivax-like. In the case of VK247, we weren't able to get this protection initially with the policy. In the previous slide, you saw that VK247 was with the lowest antibody titer, less antibodies compared to others, showing that the protection depends on the antibody response. So just to conclude, we tested another adjuvant that was Montanide 160. It's a very powerful one. And by using this adjuvant, we were able to see the protection with this meaningful difference in this pre-patent period, showing that we were able to induce protection with this protein using this very strong adjuvant. That in reality, although it's very powerful, it's producing many side effects. So we decided not to continue with the Montanide studies. So these results, the protection results, were repeated in a consistent way. Here you may see several works we developed changing the adjuvant, combining different proteins, CS with blood stages. We reproduced the data many times. We considered that we gathered enough information so that we could pass to the non-clinical studies. For that, we need the formula according to the best practice in lab, which is a big bottleneck in Brazil, that we get the certification to produce pilot batches. And basically, we established this partnership. We hired the services of the University of Nebraska, and basically they used our protocol. As you've seen, we have the growth in vials, and they used our protocol to use the different fermenters, and also we had this SDS page in the Western blot, a protein with 95% of purity. So this production was used for the validity of the analytical tests. This validation is analytical. It's an analytical stage that is needed in a way that we are going to establish the analytical methods to analyze the downstream lots, to understand if It's going to be stable, reproductive, so the batches were used for the analytical tests and also for the preclinical tests on safety and toxicity. So until this point, we explored immunogenicity, but from this moment on, we started to study toxicity, and basically these works are going to be shown by Ricardo Basinelli on the third day, and he's going to show everything that was done at SCT vacinas from our initial studies. So, a critical question remains: which is the adjuvant that we are going to use in clinical stage? This is a very important decision. We must take into account the power, but also the costs, thinking about scaling up in the future. Since the Poly C was working very well, but it's not licensed for human use, we used Poly, the one by Down-Covey, LC. It's also an R3 agonist that is not broadly licensed, but it was tested in millions of people. It's used as cancer therapy, so it wouldn't be hard for us to get this authorization to use this adjuvant. It was available through Oncovie. And at the same time, Ricardo Gazzanelli has developed this adjuvant called CT-BAD, that it was developed for SpinTech at the time in the test phase. It's a simple solution, oil and water, and it's very similar to MF59, which is a licensed adjuvant for human use. So the idea was to understand what was the most powerful one, this adjuvant that would induce T cells, or this antibody adjuvant, or this mixed response using both adjuvants in combination. So we had the same scheme of the challenge assay with the skin at Pasteur Institute, and we tested poly-C by itself, CT-FAD by itself, and the combination of both. As you may see here, the lowest levels of parasites were found using CT-VAD alone or combined with Hilton Al. The two lines here in red and pink. And the controls, of course, all of them were infected, and only Wilton All get a low protection, as we show here in the survival curve. So we had CityVAD alone protecting 83%, the combination of the two 66%, and Hiltonol alone 50%. So that was a crucial data to make the decision on what adjuvant to use, and the data showed us that CityVAD was reuniting all the features to compose this formulation. So here we have the summary. We have several other assays, but it's just to call your attention that the assays were repeated exhaustively. So here you see each one of the adjuvant tests: poly-IC, montanide, alidrogel, and the last ones we involve CT-VAD. Basically, that comparison that I've shown before, but it was tested the comparative with mice, and after deciding for CT-VAB, we used mice, and we tested immunogenicity and safety, and also in non-human primates. And we used this adjuvant for these tests. Later on, We got to 100% protection in the last test, and probably Ricardo is going to show this work that was submitted. The next step would be scaling up production for human use. So we had this GMP production in partnership with the University of Nebraska. I won't delve into details, but it was an upstream and downstream protocol, basically reproducing the data that we had obtained initially. We were able to get this pure protein with an excellent yield. So the formula that we are going to work with is called Vivaxin. So this formula is the chimeric protein that is more stable than the liquid form to be resuspended in the moment of use with the oil and water adjuvant, CDVAT. This partnership of is for veils is made with Cristália. So this is the journey from initial tests in my lab. Later on, the partnerships were extremely important until we get to the stage of regulatory approval, and we are waiting for this approval to start with uh, phase 1 tests with humans. So I'd like to reinforce the partnership with CT Vacinas because we don't have the best practice of lab in the academia, so we had to go abroad. But from this stage of characterization of IFA, all the analytic part of the product with immunogenicity tests reproduced and safety tests in mice, rabbits, monkeys. From then on, CT vacinas had an essential participation, and if it weren't for them, maybe we would have to interrupt this work. I won't delve into details because we have a poster on that, and we are now starting with this study on mRNA vaccine with the that is going to be presented by a postdoc student, and we are going to produce a RNA with blood antigens, blood stage antigens. They are candidates to vaccines, and the idea is that we can test this multi-antigen vaccine or an heterologous one. So we had the preliminary results of the PVCP-CSP protein with eukaryotic cells and other details you may see at Dennis poster. So the conclusion is that we have this Vivax malaria vaccine candidate based on a chimeric PVSCP construct incorporating sequences from the 3 major repeat variants in safe and well-tolerated in preclinical models, elicits strong immune responses, and confers robust protection in mice, supporting its advancement to phase 1 and 2 clinical trials. I'd like to thank the team from USP, my colleagues here, Silvia Buscardin and Daniel Bartieri, and all the students who participated, especially Dr. Ari, a postdoc student, and the technical team, my collaborators from Porto Velho, Maísa and Délio, the collaborators from Oxford University. We had the protection trials, Rogério Aminio, a collaborator who is very important, who has been helping us in this crucial stage, and Pasteur Institute, and the work we developed with Origin Brazil to test Policy LC. So I'd like to thank the CT Vacinas team, coordinated by Dr. Ricardo Gazzilelli, Dr. Santuza Teixeira, Dr. Helton Santiago, who is going to conduct the clinical trials. He's the clinical director, and the other researchers from the Vaccine Center, and the postdoc students who participated in scientific experiments and also in the regulatory part, which is a lot of work, and the funding agencies. Thank you so much.
De praxe, as conferências não são
As
seguidas de
usual, the conferences do not have this Q&A session, but I'd like to thank Professor Irene for her brilliant presentation, and I wish her all success as a tropical disease doctor. I'm working in transmission areas and mining areas, the greatest gold extraction region in the municipality of Itaituba in Brazil, and I've seen in a living way how important it is that we advance in this direction. Congratulations, Professor Irene. I wish you a lot of success.
Bom, me incumbe aqui a presidência
Now
da
I should go on celebrating with this plaque our honors to Professor Irene Soares for the opening of this conference, this National Malaria Research Meeting.
Okay, so after this brilliant presentation, let's go to the next section. So what I'm going to do now is to invite the speakers to join us on the floor. And so the first one is Professor Georges Nono from Institut Pasteur in Paris. The second speaker is Professor Joana Carneiro da Silva from the University of Maryland. And the third is Dr. Thais Nóbrega de Souza from Fiocruz, Minas Gerais. You can probably, yeah. And so let's start. Where is Thais? I can see her. Oh. Oh. Daniel?
We are.
So while we are waiting for, okay. So I guess we can start. So Thais will join us very soon. So let's start. Let's start with the first talk. So you probably all know Professor Georges Nono. He is a biologist and who got his PhD degree from the University of London. He has been a principal researcher at Institut Pasteur in Paris. And I'm sure everyone working on malaria knows Georges Nono for several reasons. He has been active in several areas in malaria biology, but most of us from my generation first heard his name because he developed the first nested PCR method for malaria diagnosis, and everyone in my generation used this method in both laboratory and field studies. It's a great honor to have Georges Nono here. He's going to talk about genomics, which is not exactly his main field, but as you see, he's not only an excellent scientist, but also a wonderful speaker, so it's a great pleasure to have you here. Thank you.
Well, thank you very much for inviting me for the third time to attend this conference, which I enjoy very much, and I'm very honored to be here. I'm maybe a little bit more mitigated about being arm-twisted to give a talk on a subject of which I know very little about, but I hope that it won't be as challenging for you as it has been for me to do this talk. So I hope that this is the... So first of all, I have a quiz. Can anybody tell me which year this is? No? 1977 is the year. Why is it important? That's my first year at university, but that's not the point. The point is that 2 articles were published in that year, both of them in PNAS, showing how to sequence DNA. For the first time, people had a method to sequence DNA that was easy, because there were methods before, but they were very complicated. The Maxam and Gilbert method is a chemical method, whereas the Sanger sequencing method is a dideoxy sequencing method, and indeed that's the one that has actually went on to generate the first genomics. So I will now start with my talk. So this is going to be on the rise of the genomics, and I'll remind you, I will take you back to a time far, far away where malaria was threatening the galaxy, I mean the world. Here we go. Some of you might remember the disappointment of the Malaya community in the 1950s and at the end of the 1960s, because that marked the end of the WHO Global Eradication Campaign, which was only partially successful. In other words, it was a failure. And Malayologists were quite despondent, and actually the research in Malaya had dropped down considerably during this time in the 60s. But technology was coming here to the rescue. First of all, with restriction enzymes, Paul Berg managed to generate the first recombinant DNA. So for the first time, people could actually manipulate DNA. Second, Milstein and Kohler have developed monoclonal antibodies, and for the first time we had probe in which to kind of detect specific molecules. Much more important for the Malaya community, in 1976, P. falciparum was put in culture by Jensen and Traeger, and you can see Ruth Nussenzweig talking to Jensen over here. Now, with these 3 technologies coming together, malaria researchers kind of became much more optimistic and started the era of what I would call the vaccine era. The chase for the vaccine started in the late 70s because they could now potentially identify, clone, and use the proteins that they would consider as vaccine candidates. And indeed, the first vaccine candidates appeared in 1918, that was the CS. But that's another story. It took 40 years for the first vaccine to develop since that time. So, DNA sequencing came exactly at the right time, because with the ability to clone, one needs to be able to see what the sequence that we're cloning, and without that, it would have been very difficult. Now, in those times, the late 70s and 80s, whatever your religion, you had only one Bible in a molecular biology lab. This is molecular cloning Maniatis. For those of you who worked in that time, they would probably have, this is my own copy of Maniatis photographed. It looks in good condition, but believe me, it has been much used. Now, cloning at the time, for those of you who are born after the 1990s, was a question of identifying a protein. You had to purify it, you raise monoclonal antibodies, you get genomic DNA. The easy part was getting genomic DNA, because now a parasite could be grown. You construct an expression library, not always easy. And then you screen it, you pick a clone, and lo and behold, you sequence. And once you get the sequence, you can do recombinant DNA, and you can start vaccinating mice and people and monkeys and whatever you fancied. That process usually takes, at the best of time, 6 to 8 months. At the worst of time, for instance, for the Vargenes, it took 12 years before from the time they identified the 250 kD to the time they had a first Vargene sequence. So sequencing is not sequencing. Cloning was not very easy, and to get the sequence of your gene of interest was sometimes a very complicated process. Now, sequencing was evolving very fast. So I can show you that the first ever sequenced DNA was in 1968. 12 base pair they sequenced, made a Nature paper. Wonderful. Now, this improved a little bit by a little bit, and then the first, I think this is the first, I'm not accustomed to this. The first one here is the 5X174 genome was sequenced by the Sanger method in Sanger's lab in about 6 or 7 months. And with time, with improvements, and I'll go through the improvements, by 2000, 2001, 2002, 3.5 billion base pairs of human genome was sequenced and published. So the evolution of sequencing, as you can see here, was logarithmic. The way they can. Today, we have about 34 trillion base pairs from 580,000 species in gene bank. And that's improved and increasing at the same very fast. So, what made this kind of advances occur? The remarkable technologies of sequencing. I would remind people that, you know, if when you did sequencing in 1982-83, you had an 80 centimeter acrylamide gel that was thinner than a paper, in which you had to run autoradiograph. It took 2 days, and if you were lucky and you didn't break the gel, you'd get about 250 to 300 base pairs, which you had to read and annotate by hand. With the development of fluorescent probes, and eventually with the development of capillary gels, now you can have a machine, you put it on, and you get a few thousand base pairs in a day or per run. And if you were Greg Venter, you buy 200, 300 of these machines, put them in a big room, and you generate as much DNA, as much sequences, which is why with this technology, they managed to get the human genome. Now, the human genome took about, I would say, about 8 or 9 years of work by many, many groups, and it cost approximately $2.7 billion. Now, that was a lot of money in those days, but that's the genome. Now, from the 2003, new generation sequencing occurred, and I'm not that technologically motivated, but they kind of remarkable advances, which I can't describe. But with the Illumina and eventually the ion torrent sequences, you could generate thousands upon millions of base pairs in any one run, all of them very short stretches of DNA, which then had to be assembled. There is now a third generation sequencing, and which is the PacBio and the Oxford Nanopore, and actually at the latest nowadays you have this little thing that is basically smaller than your mobile phone, which can generate gigabases of sequences on the desk, on the bench. So it really is remarkable. I dread to think what the fourth generation is going to be, and I hope I live long enough to see it. So, Plasmodium genomes. The first one, 2002, published in Nature, happened on the same year that they published the genome of humans and the genome of Anopheles gambiae, although the three haven't kind of been mixed together so far. Maybe they will one day. This took about 4 to 5 years of work, maybe 5, 6 years of work, about 4 groups, and many, many billions of dollars to generate the first draft of the genome. It's now still being corrected and improved and all of this. It took another 6 years before we got the genome of Plasmodium vivax and Nullenzweig, appeared in the same paper together. People were happy to publish them together. And eventually, a few years later, the first draft genome of Plasmodium cynomolgi, a parasite that is used quite often in experimental work. And by today, things have accelerated. We now have the genome sequences of all the parasites that infect humans and all the parasites that are zoonotic and potentially zoonotic or actually zoonotic, except maybe Plasmodium innui, but I'm sure that this will be sequenced soon. And actually, we don't have genomes of each one of these parasites. In many cases, we have thousands of genomes of different isolates from each, and I'm sure this is going to increase more and more. Now, this is generating a lot of data, and there's one crucial requirement when you generate so much data. First of all, you have to be able to assemble the genomes. To do that, you have to kind of put the pieces of DNA together. They have to put the big pieces together that they call contiguous sequences. And then, if you're lucky, you get it into chromosomes, and then you can have a chromosome architecture and makes lovely pictures in the publications. Second, you need some way to decide what is a gene, how many genes there are. Maybe try to identify what functions there are by translating them electronically, and then maybe find out promoters, microsatellites, whatever you're interested in with respect to DNA. And finally, with that data, you can then approach this kind of growing list of omics that you can do, transcriptomics, proteomics, lipidomics nowadays. Everything links back to having a genome sequence over there. Now, this is all very well, because you have access now to terabytes of sequences, terabytes of data from the genome. But if you don't have a dedicated bioinformatician, there's no point starting. I've known in many labs where people have a terabyte of data, 10 million, 20 billion DNA sequences, but they can't find an informatician who can actually put them together. So it is very important to have a bioinformatician before you embark on doing genomics. If that's one message I give, please, that's the most important one. Secure the bioinformatician. He's, sadly to say, more important than the people who do the sequencing. He's the person, or she's the person, who is going to be able to analyze the data. And if you can find a bioinformatician who understands malaria, then you hit the jackpot, because many of them don't understand it, and their analysis is sometimes meaningless. And we, as a scientist, know so little about bioinformatics and the methodology that they have, we can't even tell them whether they're doing something right or wrong. We just get the data and generate supplementary data that are wonderful to see, but are usually incomprehensible. Anyway, with the genome, I'm going to wind down quickly, we had a new hope. The malaria community in the 2002, 2003, the genome was everything. Those who sequenced were heroes. They were kind of giving the Nussenzweig talks and the Lou Miller talks. And they were the ones who gave all the talks, because they were hoping that, things are now going to improve. Now, the best improvement, immediate improvement, is that you had gene on demand. If you wanted to clone, you just dial up the gene. You just open your computer, go to the database, say, I want a gene of this. If it's identified, you get the sequence immediately, and you don't need to do the cloning. Or you can see whether there's homologs in different species, and you get the genes. Cloning became a matter of, one afternoon on the computer. Drug discovery and vaccine development were touted as the things that are now going to be solved by the genome, availability of the genome projects. We're still waiting for that to happen for these two. I don't know of many drugs that have discovered by genomics, nor any vaccines that have been developed by genomics to date. But maybe with AI, this might actually become something that will happen. The things that were useful, where the genomics gave results were functional studies and epidemiology. And with these two, one would hope to actually get the Holy Grail, which is understanding how the parasites and the host combine together. Now, exploiting the genomes with respect to the epidemiology generated with availability of the genomes, a large number of genome-wide associated studies became possible. These require big computing, little brain. To try to identify clinical markers, try to identify drug markers, try to identify antigens, the first and the third yielded nothing so far, mainly because of it's difficult to do a clinical GWAS analysis. People don't realize this, they still do them, but they haven't yielded anything, in my opinion, as yet. The drug studies were very useful because they managed to identify point mutations that were important in drug resistance. They haven't identified new drugs as yet, but they've made it easier to identify why a particular protein becomes resistant to a particular drug, which is a most important problem in malaria nowadays. Population-wise, it allowed us to give the parasites a passport. So you can tell now you have a pre-falciparum of South African origin, of Ghanaian origin, of Thai origin, you know, so we can introduce a bit of racism in malaria and decide that, you know, whether parasite is an intruder or a foreigner or is your local parasite. This has been done for falciparum, for vivax, and I know that done for other things. It does allow you, if you suddenly have a person comes in an area where there was no malaria or has malaria, you can maybe identify whether he acquired it in Honduras or acquired it in Senegal. Whether that is useful, sometimes it is, but sometimes it's just nice to know. It has also been used a lot by phylogeneticists and to understand the evolution of malaria parasites. So, for instance, in a much recent time, with more and more sequencing, we can start putting as just a, I don't know what I can, sorry. Ananias Escalante generated these kind of big tables which tells you whether the parasites of rodents came before those of falciparum, whether falciparum evolved from which parasite or not. You can also use it, as has been done here, to look at Plasmodium simian. For instance, in one study, Plasmodium simian from Brazil have all linked up to, they were closer to the ones from Mexico, which means you can blame Cortez and ask compensation from the King of Spain. But unfortunately, Marcelo has done another study which shows that actually, no, they come from Brazil. So, you know, lucky King of Spain. There you go. So this has been useful. And one has to say that the more data you have, the better the results that you have there, and that the results obtained today might, with a bit more data, with few more genomes, with a new method of analysis, because these analyses are quite complicated, you know, mathematically and statistically, you might actually find that the results are completely different. So whatever you see that looks very beautiful, maybe in a few years' time, it will not actually be, it will be that. The first phylogeny that was done, they said that P. falciparum evolved from chicken malaria. So there you go, we've evolved from them. So transcriptomics is also another field that is very important and has generated a large amount of data. This is the first transcriptome done in the Aluisio's lab, and the people who work with him are still generating transcriptome from everything they put their hands on. It gives you an idea, it gives you a kind of a map of what is being transcribed. How useful it is very difficult to say, but it is very nice to be able to know whether the gene you're looking at is transcribed early or late. And today, with single-cell technology, you get these beautiful maps of which gene is being transcribed at which stage of the life cycle, and even within the erythrocytic cycle, if I can get it there, whether it's transcribed in the trophozoites, in the late stage, in the gametocyte. So you can actually have a map of which gene is transcribed there. But having a map is one thing, and understanding biology is another. But it's good to have the map. And at least if you have a gene of interest, you can actually at least place it where it is. Therefore, you get lists of genes of potential interest, but it's only potential interest. And we have a lot of publications which just list what is potentially interesting. So, the technology that enabled all this data, however beautiful and useful it is, to start actually yielding results is genetic manipulation. First time in 1993, transiently. Second, stable transfection was achieved in 1995, and the most important thing, and quite important, is the fact that in 1997, people for the first time tagged a protein with GFP. So you can now visualize a protein with a microscope, visualize where it is, where it's going, and what it's doing. Now, this allowed us to have, and the technology for modifying the parasite genome is becoming quite remarkable and quite incredible. It's more and more complicated, not complicated, but it yields better and better results. You have a much more control about what you can make your parasite do. You have inducible markers, you protein tag them, you have gene swapping, you can go put the gene from one species in another and see what happens. It's kind of Frankenstein-style studies, but they're very useful. And that allowed us to have a lot of functional studies. I'm not going to go through the list of genes whose function has been known, because if you knock them out, what happens, what the phenotype, and what it is. And in my opinion, one of the most exciting development is in vivo imaging of parasites. And I will finish with three films. Oops, I don't know how to get back. Can you please? So these are films of sporozoites coming out of a proboscis of a mosquito. There you go, they're kind of colored in red. A person who's actually a virtuoso of doing these sorts of studies, exploiting tagging and in vivo imaging, is sitting here in the audience. Rogerio Amino is here, and these are the last two films are from his lab. One of them is sporozoite trying to find a vessel in order to go in, and the last one is a sporozoite getting into one of the areas of the liver and trying to penetrate into the hepatocytes. Now, seeing is believing, and one image is usually worth a couple of articles of speculations. So I think that this is going to be provide a lot in future. But I will remind you that in vivo veritas, whatever you see in culture, whatever you see in your beautiful images, you have to end up going and ask in vivo what is happening. And sadly nowadays, we're restricted to mice, which are not the ideal model, to macaques, which are a very good model but very expensive, and humans, which are a very cheap model but very limited in use. There you go. So, from terabytes data to parcel of knowledge, this is what genomics should give us. And hopefully you have to be able to exploit all of this, but we're limited by imagination, of which I hope they're plentiful, and money, well, unfortunately, much less than one would hope to have. So thank you very much for your attention, and no difficult questions.
Okay, thank you. It's good. I guess we will have the questions at the end, right?
Right, no problem. Thank you.
So thank you again very much, George. And we will have the questions at the end. So we do have time for the questions, but I think we can combine questions to all speakers. I think it's more productive. Okay? So our next speaker is Professor Joana Carneiro da Silva from the Institute of Genomics of the University of Maryland in the US. Joana obtained her biology degree from the University of Lisbon in Portugal and her PhD degree in genetics from the University of Arizona in the US. She has been one of the leaders in the area of malaria genomics. She is the second author of the famous Plasmodium vivax genome paper, right? And it's a great pleasure to have Joana here. She is not only an excellent scientist, but also a great collaborator. So the floor is yours. Thank you very much.
Thank you. Bom dia a todos. Good morning, everyone. First of all, I would like to thank the organizers for the invitation and the opportunity to present our results, our recent results on this theme here. Before I get there, I would like to note that while everybody here today celebrates Malaria Day, 25th of April is also a very important day in Portugal. It's a day that the Carnation Revolution, so I'm theme dressed today. So the Carnation Revolution led to the ousting of the fascist regime in Portugal 52 years ago, so in 1974. So I'm also doing resistance today. So it was a great introduction that we just heard for my talk too. The falciparum genome, the Plasmodium falciparum 3D7 genome was published in 2002, as we just heard, at the time where the number of deaths from malaria was peaking. Sorry, I'm trying to figure out this thing. The publication was the result of a consortium of several organizations and institutions, including 3 sequencing centers, one of them being TIGER, the Institute for Genomic Research, that I joined in 2002, just as the falciparum genome was coming to fruition. Then I participated in the genome papers of some rodent malaria parasites, and then later in the Vivax genome paper. And here we see Malcolm Gardner and Jane Carlton, who led the effort from Tiger. I think we were sequencing 4 of the 14 chromosomes. So in 2002, we have the 3D7 genome. We know now that it's composed of 14 nuclear contigs or supercontigs, chromosomes, that they were ordered or they were named according to length from smallest to largest, and that is overall conserved in all strains that we have sequenced to date. I'm showing here a few examples. And we also have 2 chromosomes, 1 each in the mitochondrion and the apicoplast. And soon after, a few years later, there was a meeting at the Broad Institute, supported by the Wellcome Trust, where the goal was to come up with a consensus of what next steps might be now that we have the genome. What might we, or what are the most pressing next steps? And sort of these broad 4 areas were outlined: the identification of disease-causing features, the genetic basis of parasite phenotypes, some key genes or gene products that are important for the life cycle of the parasite, and the goal of making these genomics resources available to the community. I would say that for the most part, as we will hear throughout the next 3.5 days, I would say that goals 1, 3, and 4 have produced amazing results. I would say that perhaps the point that is lagging a little bit behind, and it's the sort of the focus of my talk today, is the understanding of the genetic basis of especially complex phenotypes, and in particular, what types of information can we gather from the multitude of genetic data, genome-wide data that we have that points to genetic variation. What can we infer from that genetic variation in terms of phenotypes of interest? In particular today, I'm going to focus about these 2 topics that we're pushing forward now. One of them, as we just heard before, we know that there's a tremendous amount of genetic variation that has a geographic component to it, and another, but we don't necessarily know what functional impact that variation has. And a second point is genetic variation in known coding regions. So just very briefly, first in terms of the relationship between genetic variation and geography. So early on, Olivo Mioto and his group showed that there were differences between continents, in particular Africa and Southeast Asia. And then later, a student in my group, Kara Moser, also sort of showed nicely with this picture that that variation is present not only just between Africa and Southeast Asia, but clearly between the Americas, Africa, Southeast Asia, and Oceania, represented by Papua New Guinea there. And in addition to that, even within Africa, there's differences between East and West Africa. Later, more recently, Tony Clark and his group then have used this kind of rationale to develop a machine learning to characterize strains by geographic origin. But what we don't understand is that whether this is just simply the result of a balance between mutation and drift that leads to isolation by distance, or whether in fact there is an adaptation component to this type of geographic differentiation. Now, on the second topic, so I'll come back to the previous topic, but on the second topic, okay, now we have the genome of the clone 3D7. It was cloned from NF54, likely from West Africa, and we know that about 53% of the genome encodes for proteins. And that is where the vast majority of research has focused, is this protein coding component of the genome. And we know relatively little about the functions of known coding regions. One thing we do know is that it's incredibly AT-rich, even more AT-rich than the average of the genome, which makes it particularly difficult to study. We also know that there's hundreds of thousands or over 100,000 microsatellites spread throughout the genome. These microsatellites, there's some evidence that might be overrepresented in promoter regions. And we also know that these microsatellites mutate at a really fast rate. So why would you have some highly mutagenic sequence in a promoter region? So is this just the fact that selection is not strong against these types of rapid mutation patterns? Is it not strong enough to be eliminated by selection? Or is it that there is some advantage to some of these microsatellites? So then a couple of years later, I saw this really interesting talk by Zibi, where he showed from his group that there's a gene, PF-CYP19B, where a promoter region that has a microsatellite in it, so variation in the length of number of repeat units actually leads to, that we see here, leads to differences in transcription intensity in the downstream gene. And so once the number of repeat units deleted relative to 3D7, that's shown in pink, was larger than about 6, then the gene downstream increased, then the transcriptional output of the gene was increased, and that increased also drug resistance phenotype. So then the question is, well, oh, sorry, there's a typo there, but is this PF-CYP19B an exception? Or is it that microsatellite length might in fact be advantageous? The presence of microsatellites in promoters might in fact be advantageous in under some circumstances. So to address this, we decided to generate several different genome assemblies that were geographically representative. So we generated assemblies from strains collected in West Africa, East Africa, and Southeast Asia. This is largely work by Kara Moser and Kit Bivedi. And the reason for doing this again is just studying these types of variations, especially in non-coding regions, very AT-rich, is really difficult with short-read data, lots of inconclusive read mapping, and incorrectly inferred SNPs. And so we decided to go at it with de novo genome assemblies. And so I'm going to just mention briefly variants at 3 different levels that are pertinent to the discussion. So one of them is variants at sort of a large scale, so large structural variants. We can see from the results over here that so we're plotting the length of genome assemblies from different geographic regions. And you can see that based on these strains that we sequenced, genomes from Africa appear to be slightly larger than genomes from Southeast Asia. And one of the things that we have discovered so far is that, for example, several large-scale rearrangements shared by several of these strains, even when they are from different countries, explain some of those differences and explain also the different frequency of some of these members of multigene families in different geographic regions. So some of these very large-scale deletions that can, or expansions that can get to several dozen kb justify some of these differences. There are also small structural variants that are potentially of interest in the context of geography. So what I'm showing here is an alignment of a region, an intergenic region, or a region that we call inter-CDS. So between coding sequences, we should... so here, each of these lines, there's about 20-some, 30 assemblies lined up. You have on top in yellow 3D7, then in oranges West and East Africa, and then in red Southeast Asia, and finally at the bottom we have outgroups Plasmodium prefalciparum and Plasmodium raikkonaui. And what we see is that there are some differences that are fixed between strains that are associated with geography. So in particular, these region here, so you see downstream of these gene in chromosome 10, PF3D7, 10, 174, the promoter is completely different in Africa relative to Southeast Asia, and in Southeast Asia, Southeast Asian strains retain the allele present in the outgroup. So supposedly the ancestral allele, but the promoter region has a very large deletion in the African strains. So what are these structural variants doing? How do they contribute to phenotypes? And finally, even at the single nucleotide level, there are important differences that it's key to keep in mind. So what we did here, this is sort of an exercise in bean counting, but what we did here was we picked several hundred strains from West Africa, East Africa, and Southeast Asia, and we mapped them each to references from West Africa, so PF3D7, a reference from East Africa, MAL31, and a reference from Southeast Asia, from Cambodia, PF657. And what we show is that when you map West African samples against these three references, they map best to PF3D7. But if you now map East African samples to these references, they map, you recover most genotype or most positions in the genome to genotype if you map against an East African strain, and likewise if you map Southeast Asian samples to the three references, you recover most genotyped sites when you map against a reference, a geographically relevant reference. So it's really important to keep in mind that every time you map short-read data against a genome assembly, you are going to be dealing with ascertainment biases, and it's really important to keep that in mind. Those differences may appear small, but you can see that this is a number of genotype sites. We're dealing here with over a million genotype sites, and so some of the differences you see here are of the order of 50,000 SNPs. So these, not 50,000 SNPs, 50,000 genotype sites. So you can genotype an additional 50,000 genomic sites, and some of those might contain your causative SNPs. So it's really important to keep this in mind. What might the next steps be? So we've built now a pan-genome composed of genome assemblies from several different geographic regions, and based on this now we will be able to understand, again, these results are based on just a handful of genome assemblies, but we have thousands of short-read data sets, so we can now map those short-read data sets against the pangenome and figure out what is the actual frequency of some of these structural variants in different geographic regions. So just a few more slides now on what is happening in the non-coding regions, and a lot is happening in non-coding regions, so I'm going to focus specifically on that question of microsatellites in promoter regions. So this is work by Yiwei Yang in my group. And some of the questions that we started with are: how common are microsatellites in these promoter regions? How often are they variable in length? And does this type of variation have a phenotype? It might be that CYP19b is an exception. So what we are, the way that we decided to go about this is focusing on inter-CDSs, so regions that include the UTRs plus the intergenic region. As expected, those inter-CDSs vary in length, depending on whether they contain two promoter regions, so 5' inter-CDSs versus one stop codon, so one 3' UTR and one 5' UTR, or two 3' UTRs. So we know that these regions encode important sequences for, that are likely involved in transcriptional regulation. So in order to try to assess, okay, where are these promoters, where are the, do they contain microsatellites or not, we needed to go through several steps. So use 3D7 to identify those genes that have UTRs annotated, then identify the promoter region, then picking up on all the other genome assemblies, they needed to be annotated to identify the location of the genes, determine how reliable that annotation was, and then identify the orthologous inter-CDSs between genome assemblies, and finally extract those orthologous inter-CDSs, align them, and identify microsatellite variation. So the first step is more or less trivial. The second, third, and fourth are not, but Yiwei has done a lot of work on this. So what I'm showing here is an alignment of a variety of assemblies. And the orthology here, the orange lines show connect orthologous gene sequences once the assemblies were annotated. And you can see that for the most part, assemblies are very syntenic, and therefore the inter-CDSs are going to be largely orthologous. How good was the annotation of those genes? Well, the annotation is pretty good. What I'm showing here is that for several of these genome assemblies, one caller per assembly, Yiwei identified how many genes differ in length for the annotation between that particular assembly and 3D7. If the genes are correctly annotated, in particular the start and the stop, the position of the start and stop codons, then the inter-CDSs next to them are going to be correct. And what you see here is that close to 95% of the genes do not differ in length between 3D7 and the other strains, meaning that the annotation is likely correct, and therefore the inter-CDSs that flank them are also likely correct. So about 98% of the genes fall sort of within this 5% length variation relative to 3D7. So given that this seems to be solid, then what Yiwei went through was a series of sieving steps where she identified which of these inter-CDSs have orthologs across all the different assemblies, and of those, how many have microsatellites in the promoter regions, to then be able to determine are those microsatellites variable in length or not. So in the end, she so far identified that about 600 of these inter-CDSs have short tandem repeats in promoter regions. those short tandem repeats, we're limiting them to di, tri, or tetranucleotide repeat units. And almost all of them have variation. So 613 out of the 642, they have length variation. Reassuringly, the CYP19B gene came up in her screen, and so what we're showing here is, so here you have sort of the actual intergenic region and the promoter that has length variation across these assemblies. So this display is very much like I've shown before, with 3D7 on top, then West and East Africa genomes, and then Southeast Asian genomes. In teal here, you have the 5' UTR of CYP19B going that way, and in green, we have the 3' UTR of the upstream gene that's transcribed from left to right. So this is a 3', 5' UTR. You can see the 5' UTR is really, really small, and it's basically really that repeat, that A and T repeat. So next steps for EWAY are to culture all these different strains that you see here. She is going to use a variety of different conditions, and then assess gene expression in genes that have these variations in promoter regions to see whether or not CYP19B is an exception or the rule. Reassuringly, one thing that she also looked at was, well, but are these microsatellite repeats really likely to impact the expression of downstream genes? And if they are, you might expect that these microsatellites might be absent in promoters of genes that perform really critical functions. So she looked at about 2,500 genes that do not have short tandem repeats in their promoter regions, and determine what are the molecular functions of those genes. And sure enough, you will see that most of these functions are related to ATP binding, or nucleotide or ribonucleotide metabolism, so really the basic functions of cells. So in fact, this is evidence that suggests that, in fact, this particular type of repeat in the promoter region is likely to change the expression of downstream genes, and therefore, through evolutionary time, selection has eliminated these short tandem repeats in upstream of genes that are really critical. What did I want to say here? So there's a variety of questions that we will be, once we have the experimental data, we will be able to determine, in particular, which genes undergo sort of a similar mechanism as CYP19B, and what functions are those genes involved in. So in concluding, I just wanted to stress that much remains to be discovered about the function of the genome or genomic regions in Plasmodium falciparum, that many valuable hypotheses can be generated just by comparing existing data that then obviously need to go and be tested experimentally, that awareness of reference bias in genotyping is really important, and that much remains to be discovered in regarding the function of regions that do not code for proteins. In particular, for example, UTRs. I'm not showing that type of data, but they are much more conserved than you would expect by chance. For example, we know that there are hundreds of known coding RNAs and so on and so forth. So just to conclude, the work I presented before was mostly work from Kara Moser, Ankit Vivedi, and Yue Yang, and I would like to also acknowledge my collaborators at the CVD, at the Center for Vaccine Development and Global Health at the University of Maryland School of Medicine, in particular Shannon Takala Harrison, Kirsten Leik, and Matt Lawrence, who have been longtime collaborators. Some outside collaborators at Scenaria, Swiss TPH, and Mali, and finally the funding agencies that funded this particular work.
Is it working? Yeah. Please remember that we will have our discussions at the end, okay? So the next speaker is Thais Nóbrega de Souza. Thais is a biologist, graduated from the Federal University of Minas Gerais, and she obtained her PhD degree at Fiocruz in Minas Gerais, where she is currently an associate researcher. So Thais, it's a great pleasure to have you here, and the floor is yours.
Thank you, Marcelo. I swear I wasn't trying to run away because I had a little delay. So I would like first to thank you for the organizers, for the invitation. It's a pleasure to be here today. And today I'll be talking about our efforts on tracking drug resistance in the Brazilian three-border region. Between Brazil, Venezuela, and Guyana. Let me just, this region. So this is a region where we have great concern regarding the rising of malaria, especially in vulnerable territories, okay? And here we are talking about in special indigenous lands, where many factors are associated with the risk of, this rising of malaria cases, including the overlap with mining zones, mining activities, especially illegal mining activities, okay? But also, these are places characterized by remoteness with limited access to healthcare and also intense human mobility. So, in this study, Marcelo Urbano's group has pointed to the mobility as an important risk factor for malaria transmission, where they identified that 1/3 of the Vivex infections were actually acquired outside the locality of residence. And here we can see that Boa Vista, which is the focus today, is the region. This doesn't work. Okay. Is a major sink for malaria in the Guyana Shield, meaning that in this region, okay, we have more import of malaria cases than export. Okay, and as I told you, this is an important risk factor for malaria transmission. Okay. And so today, our topic is related to malaria treatment. For those who are not familiar with the treatment in Brazil, for Vivex, it's recommended chloroquine plus primaquine. For the recurrences, ACT plus primaquine. And for P. falciparum, the first line treatment is artemether lumenfantrine, and the second one, artesunate mefloquine plus a single dose of primaquine. And the rationale behind this combination of drugs to treat P. falciparum is to delay or to avoid the appearance of resistance, drug resistance. But unfortunately, we have seen already the emergence of resistance, partial resistance to artemisinin, and also to its partner drugs. Okay, we here can see shown on the black arrows that the resistance has been reported for almost all drugs that are currently used And also in Guyana Shield, we have already this identification of an important mutation in the main gene, the gene that is the main determinant for partial resistance to artemisinin, which is the KH13. So it has been already described in Guyana, and also some variants to the partner drugs. So this is a big concern in this region that we'll be talking today. So all these observations motivate us to implement a genomic surveillance initiative to track antimalarial resistance markers in this region delineated here in the map, which is known as Guyana Shield. Okay, so here we see we have this. We have this border between Brazil, Venezuela, and Guyana. And this is a place characterized to be a hotspot for malaria transmission. Okay, and also an epicenter for drug resistance. Why? Because this region shows many factors, factors associated with drug resistance, including intense human mobility, also illegal mining activities, okay, and which are often associated with irregular use of antimalarials. Okay, so all these factors contribute somehow to the emergence and the spread of resistant variants, especially in this place. So, in a collaborative project initiative, our main goal was to develop a tool able to detect low-frequency variants in polyclonal infections, which are very common in malaria infections. And why this is important? Because the early detection of these variants can anticipate the expansion of these variants in the parasite population. So our approach included deep sequencing of a panel of genes, 48 genes, all of them associated with drug resistance, okay? So full length, 48 full-length genes of P. falciparum and 11 genes of P. vivax. Very briefly, the workflow included a whole genome amplification prior sequencing and a dedicated bioinformatics pipeline in collaboration with Dr. Antônio from Fiocruz, Minas. He's a bioinformatician. And here we can see that our approach achieved high depth and also high coverage for the 48 genes of Plasmodium falciparum. The high depth, read depth, means that each base was sequenced on average 1,000 times, giving an impressive sensitivity to the method, which is important to characterize this kind of low frequency variants. And also the panel had showed good performance for DNA extracted from whole blood, but also from dried blood spots, which we expect to recuperate low amounts of DNA. So this is the most important results for this part of the study, because here we see for each sample on columns that we sequenced for the 48 genes, we see this enrichment of low-frequency variants, here shown on lighter blue. And this pattern is consistent with polyclonal infections, which are expected to be more prevalent on high endemic areas as Mali on the African continent. Okay, so what does it mean? It means that depending on the sensitivity of the method, these variants could be missed and remain undetected. So the panel also showed a great performance for Vivex targets, high coverage proportion, and also high median depth. And here again, I show in a different representation this enrichment of low to intermediate frequencies, of variant frequencies. And again, here, depending on the sensitivity of the method, this could be missed and remain undetected. So, we applied, by applying this approach to a set of falciparum samples collected in Roraima state, we showed using the 48 genes, the high genetic relatedness between the Roraima samples, Brazilian samples with French Guyanese samples, okay, probably reflecting the close proximity, but also the human, the human migration in this area, in the Guyana Shield. But here I would like to highlight an important finding that we had in this part of the study. We were able to detect this variant in CRT gene, okay, and only using this approach and not Sanger sequencing. And this variant, along with multiplications in plasma epicines 2 and 3, are associated with piperazine resistance. This has been already described in high prevalence in Guyana, Suriname, French Guiana. Okay, this was the first time reported in this area. But one curiosity here is that piperazine is not a drug officially used in Brazil or in any of these countries. So the possibility is that the irregular use of 1-ACT containing piperazine that has been already reported in these areas, especially by miners, can be the driver for this selection for these variants. Okay, so this is an example of a clear example of irregular use of drugs driving the selection of drug resistance variants in this area, in Brazil. So regarding the drug that has been used for decades to treat P. falciparum malaria, artemether lumefantrine, we for the first time now are documented the emergency, the appearance of this haplotype associated with MDR1, which is a gene associated with, in this case of NFD, is related to lower susceptibility to lumefantrine, okay, and is also being seen in other areas where artemether lumefantrine has been used for many years. So this is the first time that we are documented the appearance of this haplotype. And another evidence for this selection on MDR1 is this sharp decrease in microsatellite variability around the MDR1 gene in the isolates that we have analyzed, that we have characterized this microsatellite variability along the chromosome 5, where the gene is located. So we also were interested to characterize isolates Vivax samples from the same area in order to understand if similar selective pressures could be also modulating, shaping Vivax genetic variability in this area. And here we applied the panel for Vivax. So by analyzing these 11 genes, we were able to clusterize, see the clusterization of Roraima samples with samples from the Guyana Shield area. Okay, and here we can see that they are more closely related to these samples from these countries, neighboring countries, than to those from other areas from Brazil. So this is interesting, and we raised here an hypothesis that the unintentional exposure of Vivax, okay, to treatment, more specifically to P. falciparum treatment, could be driving this genetic the selection for drug-resistant parasites or drug or variants, resistant variants for the parasite. And why unintentionally treatment? Because when we reanalyze, this is a place characterized by co-circulation of Vivax and falciparum. And when reanalyzed a set of samples by qPCR, we found a surprisingly high prevalence of mixed infections. Okay? So these were all samples initially classified as monoinfections, either by Vivax or falciparum. And by reanalyzing by qPCR, we found that actually 20% of these mixed infections were 20% of these monoinfections by microscopy were in fact mixed infections by Vivax and falciparum. And the most frequent mixed infection was Plasmodium vivax. So not surprisingly, because Vivax, we know that the parasitemias are lower in general compared to P. falciparum. And here we show that actually we had a very lower proportion of Vivax in these infections with mixed infections mixed infections with Plasmodium falciparum, as we can see here in the first part of the graph. And it's interesting to note that this the underreporting of Vivax infections somehow fluctuated or parallel with P. falciparum cases. These were cases identified by the surveillance system. Okay, so it suggests somehow that the co-circulation with P. falciparum may increase the likelihood of missed P. vivax infections. So, but the most important question here was what would be the consequences of this exposure of P. vivax to this incorrect or unintentional treatment, because all those infections were treated by were treated using the P-falciparum treatment. So what we found was also a surprisingly high prevalence of MDR1 amplification, okay, in these samples, and it seems to be restricted in Roraima state, when we compare with samples that were also analyzed from the Western Amazon. And this phenomenon has been increasing, this selection has been increased over the years analyzed here. Okay, so MDR1 is the main determinant for mefloquine resistance, okay? But also it's associated with resistance to other drugs like lumefantrine and also artesunate. But in regions that the similar phenomenon has been observed, they associated this increase of the PV-MDR1 amplification mainly with the use of mefloquine to treat P-falciparum. Okay, so Vivax being exposed to a treatment of mefloquine, but targeting P-falciparum. But we don't have a clear evidence for this intense use of mefloquine in these areas that we are studying. So here we cannot clearly underline the factors associated with this selection, nor the clinical consequences of this amplification, but we suspect or we raised the hypothesis that the use of lumefantrine can be the driver for this selection. And why? Not coincidentally, we have seen the same phenomenon now with P-falciparum in Angola, in Africa. Also, the selection of MDR1 amplifications by the use of lumefantrine, artemether lumefantrine. So we are not sure, but this can be also something that is happening in this area. Okay, but one thing is clear, it's not chloroquine, which is this driving force, because mutations in MDR1, including these amplifications, have opposite effect on chloroquine and mefloquine and possibly lumefantrine, in a way that these multiplications is actually associated with a lower sensitivity to chloroquine, okay? While gives a higher sensitivity to mefloquine and possibly lumefantrine. So, in conclusion, we are, by applying a highly sensitive panels, highly sensitive method, we were here able to review signals of selection in P-falciparum, in this important area of study. And also here we present evidence for under-detection and inadequate treatment of Vivax, contributing somehow to the emergence of MDR1 amplification. We need now to understand the consequences of this. And altogether, these observations raise an alert for the ongoing use of genomic surveillance and the early action to preserve treatment efficacy, especially in this area, this endemic area in Brazil, okay, the Brazilian tree board. So, I'd like to thank you, all the collaborators, including our students who has been working very hard in this project, and also the funding agencies, and thank you all. That's it. Okay.
So we have very limited time for questions, but I think this is very important. So if you can keep your questions very brief, I'd be pleased to have the session now. So who would like to present any question? Please go to this microphone, I guess. That's the easiest way. And you must have some microphone.
Quick question for Joanna. Nice talk about the non-coding regions. Quick question. You did analysis of the promoter sequence. Have you ever think about doing the same for intronic sequences and see like size variations and like transcription variation in the like caused by the intronic sequences?
Thank you for the question. Yes, I think there are. I'm interested in all these potentially relevant regions of the genome, introns as well as UTRs and intergenic sequences. We have a lot of evidence that suggests that they're more conserved than they should be by chance. But I have not looked into introns, and I don't have the bandwidth to do it. But I would encourage anyone who is interested in doing that. I'm sure you'll find something.
So, more questions from the audience? Come on, please, don't be shy. It's the first session. So, great. Marcia.
Thank you very much for excellent presentations. And I have a question to Joanna. And Joanna, you are working with field isolates for several places in the world. And have you considered host characteristics or polymorphisms that can direct these selection in the parasites or vector species? Thank you.
Excellent question, Marcia. I think you're right on the mark there. We do have preliminary evidence that some of the geographically relevant differences between strains from different geographic regions, that a large proportion of them has to do with adaptation to different vectors. As you might be aware, vectors are very different in Southeast Asia and in Africa. And in addition to that, yes, I do think that it's really critical to look at the contribution of host factors as well as vectors in acting on these variations. But it's only by, you need to start somewhere. So I think first sort of digging up some of these variants and trying to select them in a way. So that's why we selected things that are, or variants that are fixed for different allelic states in different continents, so that we have both a difference, potential difference in vectors, also potentially difference in the host, the human host populations. So yeah, I think there's plenty of room to explore host factors there.
More questions? Okay, so Beto.
Joanne, so this bias issue has to do really with the intercoding sequences. That's what you're saying? I'm trying to wrap my mind around this and how it affects any of our analyses. So that's just for the intercoding sequences? Because our genes, we know what they are, and so you know to align across the named genes. And I wonder if the solution also could be naming these intercoding sequences if they're consistent. And then I guess those turn up as missing data in your analysis across many genomes. Is that what happens? It's just missing data?
So I think the way to think about it that makes it, I think, fairly obvious is that Plasmodium falciparum 3D7, for example, is missing large regions that are present in other assemblies. So those regions you won't even be able to genotype, right? And while it's true that for a large extent we know a lot about different genes, the single copy genes are relatively easy to identify whether or not they're present, but then you have sort of a whole hundreds of members of multi-gene families that are present in very different frequencies in different geographic regions. And part of it is that they're missing large pieces of chromosomes, and you'll never be able to identify those if you map simply to 3D7. But yes, in addition to that, some of these genotyped, additional 50,000 genotype sites that we're able to catch, some of them will be present in these regions that are missing altogether from 3D7, and some of them are just present in really variable regions, including in some of the members of multi-gene families, and you just can't genotype, you can't identify those variants, because the reads from different clinical isolates won't map to 3D7.
Right. So then maybe the solution is not using parasites that have been in culture for decades and maybe sticking to field isolates.
There's many interesting data to dig from here, but one of the things that we see, for example, is in Southeast Asia, the strain that had the longest genome is actually NF135, which has been in culture, as you know, has been in culture in the lab for a long time, and so it has not been subjected to a lot of these more recent selective pressures from a variety of drugs and so on and so forth. So yes, there's also that dichotomy between lab cultural strains versus more recently collected strains.
Thank you.
Okay, so maybe the very last one, very brief, please.
Really, not even a question. I just want to thank Dr. Snusnu for acknowledging the bioinformatician and its importance in this whole era of genomics right now. So, thank you.
So, thank you very much once again. It was a brilliant session. We will have a very brief coffee break, but of course, I know many of you have interesting questions to all the speakers, so please don't be shy and approach them during the coffee break. So thank you very much once again to the speakers and the audience.